All right, well, last week I told you about Joshua Letterberg, who was a microbial geneticist here and a world-class scientist, one who had enormous influence on the development of genetics as a field everywhere in the world. And this week I'd like to tell you about R.A. Brink, who was a professor here for many years and was a plant geneticist. His name was Royal Alexander Brink. That name makes you think, of trucks with full of cash and guards at the door. And you might think that Royal Alexander Brink, with a name like that, was some sort of aristocrat. But in fact, he wasn't. He grew up on a farm in Canada. I take it not a terribly profitable farm. He was born in 1897, died in 1984. And he grew up on this farm in Woodstock, or near Woodstock, Ontario, Canada. It was a dairy farm. He attended a one-room schoolhouse until age 11. He says he liked that the schoolhouse says he remembers it was a pleasant environment. Now, several people who were professors in our department had the experience of studying in a one-room schoolhouse in their grade school days. Sewell Wright, for example, who was a famous geneticist, studied in a one-room schoolhouse. Brink remembers that he liked the place because he learned a fair amount, and he loved the maps that were pasted on the walls all around the room. So he learned about the size and diversity of the world while he was a student. After he got finished at the grade school, he entered high school at a place called the Collegiate Institute in Woodstock. To get to the Collegiate Institute, he had to get up in the morning, milk 5 cows, do some other chores around the farm, walk 2 miles to a train, take the train 5 miles to Woodstock. He was so tired, and he had to do the same thing, of course, when the day ended and he came home. 5 miles by train, 2 mile walk, milk cows, do some more work. He said he never got any homework done, and he didn't do very well at school. His teachers thought he was pretty stupid. They never called on him because he never knew the answers to anything. And he felt quite humiliated during his first year of school. He even flunked the year-end final exams. And he talked to his dad about it and said, I can't keep up this schedule. You have to do something for me. And his dad said, well, I'll tell you what, 2 weeks before the year-end exam, you can just devote full time to studies and you don't have to do any farm chores. So he crammed for 2 weeks before the exams at the end of his high school years, and he got firsts on all these exams, much to the astonishment of his faculty, the faculty members at the high school, and he graduated from high school with honors. So that was the Collegiate Institute in Woodstock, which he said was a miserable experience. He remembers high school with unhappiness. But he had always thought that he ought to go on and get an education and escape from the rugged life on the farm that he was living, and he entered the Ontario Agricultural College in Guelph after he graduated from high school. Got off to a bad start. This first year in high school, he contracted typhoid fever, and the doctors that examined him misdiagnosed the typhoid and thought he had appendicitis. They did an appendectomy on him. He was pretty sick before they did the appendectomy, and he was even sicker after he did the appendectomy, so he was in the hospital for 10 weeks. He developed a blood clot in his leg, from which it took him about a year to recover. He was walking around on crutches after he was released from the hospital. So he didn't do very well his first year at college, but he went back the next year, and with the help of friends, he managed to pass all the exams for the freshman year, and then he went on and got a college degree in chemistry and physics from Ontario Agricultural College, graduated in 1919. With his new degree from college, he would have liked to go on for other study, but he didn't have any money, and his hospitalization had cost his father essentially all the resources that the family had. So he went to work, and he went to work for the Western Canada flour mills in Winnipeg. That's a picture of what it looked like, I think, at the time. He was in charge of testing wheat in a flour milling operation. They produced 5,000 barrels of flour a day, and his job was to run the bread baking test to make sure that the flour that they were grinding into, that the wheat they were grinding into flour was high quality. He said that this was an interesting job, actually, and he was well paid for it, and he rather enjoyed doing it. But he realized that after 10 years or so of doing this job, he didn't see that he was going to go anywhere beyond being a tester of running the bread making test. He thought this could get rather monotonous, and that he would do well to go and get more education. And he'd saved up just enough money to think about going to do some graduate study, which he did. He went to the University of Illinois, and he took a course in genetics from a man named Detlefsen. He loved the course in genetics. He was planning to study agronomy, but he got interested in genetics as a subject. And he worked in Detlefsen's lab on fruit flies. He was doing a study on the inheritance of the variations in the frequency of recombination of genes in fruit flies. It was demonstrated that flies actually had some genetic control over how much recombination occurred, and he was doing work like that under Detlefsen's guidance. And then he wanted to stay on and do graduate work in Detlefsen's lab. And Detlefsen said, sorry, I didn't get tenure. Apparently he was not an easy man to get along with, and the faculty at Illinois didn't like him. But Detlefsen had been at Harvard before he got a job on the faculty at Illinois, and he knew Edward East, who was a professor of genetics at Harvard. And he wrote to East, Detlefsen wrote to East and said, I've got a good candidate for graduate work at Harvard. Would you be willing to take him on? And they offered him a scholarship that covered his tuition and paid $50 in addition to the cost of tuition. And he thought that was a job, that was an offer too good to refuse. He went to study in East's laboratory at Harvard, and by 1923 he had earned a Doctor of Science degree. He was working while he was in East's lab on the physiology of pollen growth, and he was growing pollen in culture. So he would take pollen grains and germinate them in culture. He was trying to find conditions under which he could grow individual pollen grains. They'd grow a pollen tube, and they'd undergo cell division, meiotic cell division, so that he could see what a pollen grain needed for growth and function. And East at the same time was working on corn, and he was working on the improvement of corn. He was hoping to make a better agricultural crop from corn. So Brink was becoming familiar with what corn breeding was all about, and when he came to the University of Wisconsin in 1923, he just got his degree from Harvard, and he was offered an assistant professorship at University of Wisconsin. And at the time, the genetics department at the University of Wisconsin consisted of L.J. Cole, who was the first chair of the department. He was hired in 1910. The department was called the Department of Experimental Breeding when Cole was hired here to start up a department. When Cole was hired and the Department of Experimental Breeding was first formed, the mission of the department was to do animal breeding and to improve cattle, both beef and milk production in cattle, by doing selective breeding of cattle. This happened because there was a member of the Board of Regents whose name was W.D. Hord, who was the publisher of Hord's Dairyman, a trade magazine that's still being published today. Hord had been governor of the state, and at the time that the department was created in 1910, he was a obviously prominent member of the Board of Regents, and he used his influence to get the university to approve the creation of this new department. But the department was 12 years old by the time Brink got here, and it still consisted of one person. It had hired another geneticist, Lindgren, for 1 year, and then he went off and started a department at the University of Iowa, leaving Cole all by himself again. Cole recruited Brink, and Brink came here and started talking to other members of the faculty in the College of Agriculture and discovered that the dean's office didn't think much of the Department of Experimental Breeding, and the other members of the college didn't think much because they weren't really improving cattle. Cattle genetics takes a long time. You know, it takes a lot of time to go from cow to calf to breeding cow again. And one cow is an expensive experimental organism, and if you want to work with hundreds of them, you find that the College of Agriculture doesn't have enough money to support that kind of research. So poor Cole, although he was doing his best, wasn't making a big impression on the administration or on his colleagues in the department. Cole, however, was terrific at building graduate programs. He contributed to the development of the graduate programs and the creation of the graduate school. He had a lot of students, and they were wonderful students, and he gave them a great education in genetics. He worked on the genetics of birds, of pigeons, and he made real contributions to pigeon genetics. They're much cheaper and faster breeding than cattle. Anyway, Cole went off to Washington for a year, right after Brink got here, to work at the USDA, and left Brink to his own devices. And Brink started talking around about, what do you think about starting a hybrid corn breeding program for the state of Wisconsin? Hybrid corn was just beginning to be a thing in plant science. And Brink had worked with East, who, together with George Shull, was one of the founders of hybrid corn breeding. So he was familiar with hybrid corn breeding. He had visited a very successful corn breeding operation in Connecticut that was run by a geneticist named Donald Jones, who's kind of a towering figure in the history of plant genetics. So Brink had a pretty good idea about how you ran a corn breeding program, and he suggested to the director of to the chair of the Department of Agronomy, whose name was R.A. Moore, that they start trying to breed hybrid corn specifically for the state of Wisconsin. It was too cold up here to grow corn very well, and they needed to try specifically to develop corn that would grow in Wisconsin on Wisconsin farms. Moore, who was busy trying to do the same thing but didn't want to use hybrid corn as a technique, said, well, I don't think it's going to work, but what the heck, let's try it. So with not very much enthusiasm, he agreed to let the Department of Agronomy work with Brink. And in 1925, Brink started a program on breeding hybrid corn for the state of Wisconsin in cooperation with the Departments of Agronomy, Plant Pathology, and the U.S. Department of Agriculture. And he had that's a mistake. I said he had a graduate student named Norman Neal work on the project, but Norman Neal was here as a postdoc when he was in Brink's lab. My fingers got loose when I typed that, or my brain. So here's Norman Neal. After a few years of cooperative work in which Brink was the guiding figure, he turned over the program to Norman Neal, who stayed with the program for quite a long time, a number of years. So Brink initiated the hybrid corn breeding program in 1923. In 1925, after doing a lot of planning and talking and making arrangements to allocate fields in the experimental farms and so forth, the work actually began. And in 1931, Neal took over the direction of the program. And in 1933, the first hybrid corn was released for commercial sale in the state of Wisconsin. So that's a 10-year period. You understand that corn, like cows, has a fairly long breeding time as well. It's a generation time that seems just infinite to people like me who are microbial geneticists. Microbial geneticists, you do a cross one day and you look at the offspring the next day. With corn, you do a cross one day and you look at the offspring 3 or 4 months later. They could grow 2 corn crops if they went down to Florida to grow some, but these people were trying to develop corn that would grow well in a Wisconsin climate. So the first corn was released in 1933. That's wrong. 1933. Richard Crabb, who wrote a book called The Hybrid Corn Makers, which discussed all of these people, Brink and his collaborators, estimated that by 1947, when he wrote this book, the collective benefit to Wisconsin farmers was about $20 million per year from having replaced just any old corn with very select Wisconsin-suited hybrid corn. $20 million in 1947 bucks was about $220 million in 2018 bucks. So that's pretty good return on investment, developing hybrid corn for the state of Wisconsin. Investment in the program, I just thought you might be interested to know that when Letterberg started here in 1948 as an assistant professor, his salary was $4,800 per annum, which gives you a feeling for about what these people were being paid for doing the work that they were doing on corn development. Prices were fairly low. $220 million is a huge amount of money compared to the amount of money that they were, or even $20 million compared to what the state and the federal government invested in this corn breeding program for Wisconsin. I just want to teach you a little science. I suspect you're all familiar with this, but this is what the corn breeding program looked like. As you probably know, if you inbreed almost anything, you get less and less fit organisms generation after generation. The more inbreeding, the less fit the organisms are. And the reason for that is that as you inbreed, you lose genetic diversity and you get more and more genes that are homozygous generation after generation. And some of those genes are going to be recessive mutants that have a significant effect on the fitness of the organism. So the more you inbreed, the weaker the organism gets. But if you cross two hybrids to one another, you get offspring that come from different backgrounds and have different homozygous genes, and whose dominant genes can cover one another's weaknesses. And you get something called hybrid vigor, that you have to try a lot of different inbred lines and cross them to one another to look for the ones that give the most vigorous offspring. But this is a way of getting plants that are better on the average than any plants that you can get by just picking the tallest plants generation after generation and using them to start the next generation. And furthermore, you get a much more uniform crop because all the inbreds have the same genotype. So if you cross two inbreds with one another, you get plants that have a uniform genotype throughout. Plants all grow to the same height. They have ears that are the same length. They have the same number of kernels on the ears and so forth. So here's the breeding plan that they used, which was developed by Donald Jones. First, they crossed two inbreds to one another to produce a hybrid, which was vigorous. Crossed that hybrid to another hybrid that was produced by crossing two other inbreds to one another. So these are little sort of feeble plants. When they're crossed to one another, they give vigorous hybrids. And when the hybrids are crossed to one another, that's called a double cross, they produce a nice, vigorous plant. Has lots of genes from independent origins, pretty uniform size. They get a lot of seeds. So this is the plan that Donald Jones developed at the breeding station in Connecticut. And this is what gave rise to these very useful hybrid plants that invigorated corn growing in the state of Wisconsin. So that was one of Brink's contributions to the state. He came to the department. He saw that there was a problem that geneticists could address. He addressed it in collaboration with other departments in the university, and he produced a product that was of tremendous economic benefit to the state of Wisconsin. So here are two inbred lines of corn, and here's their offspring, the F1 hybrid. This is what the ears look like from these guys, and that's what's called hybrid vigor. Over the years, Brink died in 1984, around there, but you'll notice that once the project started, the yield per acre of corn increased year after year until it finally had increased something like 5-fold. So that corn that was economically sort of iffy before the hybrid corn was introduced became much, much more profitable after the introduction of hybrid corn. Brink also took on a project that involved an important leguminous forage crop, sweet clover, which grew in Wisconsin but didn't grow terrifically well, and that had a problem. And the problem was that even the forage crop that they were growing in Wisconsin was bitter. And although cows would eat it if you didn't give them anything else to eat, they didn't like it very much. And furthermore, if this sweet clover fermented before you fed it to corn, if you stored the hay that you cut from your field of sweet clover and it fermented a little bit, the spoiled sweet clover was poisonous to the cows. Well, in 1933, Brink, after a great deal of effort, looking around at lots and lots and lots of kinds of sweet clover, found a sweet clover variant that was non-bitter. And he got to work with Willard Roberts, who was a student in Carl Paul Link's lab in the Department of Biochemistry. And the two of them determined that the difference between bitter and non-bitter sweet clover was the presence of a chemical called coumarin. Now, it was known for a long time that coumarin was present in sweet clover and that it was responsible for the sweet odor of sweet clover. But it wasn't known that the bitter taste of sweet clover was due to the presence of coumarin. And then Brink, working together with W.K. Smith, who was a postdoc in his lab, and then became a professor in the Department of Genetics and Agronomy, worked together and discovered that, in fact, the toxicity that was associated with the sweet clover was somehow correlated with the presence of coumarin. And then working with small animals, with rabbits, Smith discovered that if you fermented ordinary sweet clover, you got a poisonous product, but if you fermented the non-bitter sweet clover, you didn't. So the non-bitter sweet clover that Brink had discovered was apparently safe feed, whether it was fermented or not. Actually, the cattle kind of like it fermented, so that would be an advantage. And then Smith discovered that if you added coumarin to the non-bitter sweet clover and you fermented it, that the addition of coumarin caused the sweet clover to become toxic. So the toxicity of sweet clover was due to the presence of coumarin, and the fermentation process turned the coumarin into something that was lethal to cattle and caused them to develop hemorrhagic disease. Smith took over the job of trying to get the gene that was responsible for the non-bitterness of Brink's sweet clover into an agriculturally desirable strain of sweet clover. He had to do a lot of work. It involved taking cuttings from the non-bitter sweet clover, and first he had to cross the two kinds of sweet clover. They weren't, the offspring wouldn't grow, so he had to grow them by doing grafts onto other plants and see if he could grow up some flowering offspring from the cross between bitter and non-bitter sweet clover. He eventually managed, after a good many years, to produce a variety which is called Denta. You'll notice this started in 1938. It ended in 1964. But in 1964, they managed to release a variety of sweet clover that was non-toxic and that was winter hardy and that grew well on Wisconsin soil. Carl Paul Link and his colleagues then took on the job of trying to identify what was the hemorrhagic component of sweet clover when it was fermented, and they discovered that it was dicumarol, which has become a commercial product. It's both used as an anticoagulant in medicine and as a rodent poison. It's made a lot of money for the university and a lot of money for Link and for the biochemistry department. The story that has been told over and over again about how Carl Paul Link got involved with the study of sweet clover is that a farmer showed up one day with 2 buckets of blood from a cow that had died, and Link got inspired about working on this problem of poisonous sweet clover. But in fact, Link was just brought into a project that was already pretty well advanced. They already knew about the nature of the toxic compound in sweet clover, and they knew that it had to be a modified form of coumarin that gave rise to something. Carl Paul Link and his colleagues discovered what that was and developed it as a commercial product, and a very important one at that. Another one of Link's projects, Brink's projects, was the development of a winter hardy alfalfa. Alfalfa is an important forage crop. It's a perennial. You can cut it 3 or 4 times a year, and after you've cut it, it grows back up, and you can cut it again. It grows year after year. It fixes nitrogen, so it improves the soil. It stabilizes soil against erosion. So it's a terrific crop. The only trouble with it was cold. It's cold sensitive, and it didn't grow very well on Wisconsin farms. It was a great feed for cattle, and Wisconsin was becoming, thanks to W.D. Horde, a dairy state. So Brink thought that it would be a good idea to see if it was possible to breed alfalfa that would grow well in the cold climates of Wisconsin. You couldn't grow any alfalfa at all in the upper half of Wisconsin before Brink did this project. So in 1926, he initiated a breeding program to make a winter hardy alfalfa that was also had one other good quality, that it was resistant to wilt, which is an infectious disease, an infective disease, a pathological condition of alfalfa. This was a tremendously complicated program, which he took on in conjunction with a fellow named Fred Jones in the Department of Agronomy and the USDA, and with Dale Smith in agronomy and Lawrence Graber, numerous genetics graduate students. And they managed to find a winter hardy alfalfa, and then by crossing it to other alfalfas, to develop a sort of Wisconsin-specific kind of alfalfa that would grow well, survive through the winter, grow up well in the spring, produce a lot of cuttings during the summer, didn't develop wilt. Vernal alfalfa was the name of the alfalfa that they finally bred. It became the most commonly used alfalfa in Ohio, Missouri, Iowa, Minnesota, Michigan. It's also grown in other northern states and Canada. On a vacation to Canada, after vernal alfalfa was released, he went to visit his family farm, which was no longer owned by Brinks but by somebody else, and discovered that they were now growing alfalfa on that field. That his family's farm was now essentially an alfalfa farm, and it was all vernal alfalfa. So he felt this kind of personal pride that he had repaid his home farm by giving them vernal. In 1970, 21 million pounds of vernal seed were sold. It's inconceivable to me that you could even collect 21 million pounds of seed. But anyway, that's the way it was. It's still One of the most common alfalfas used in agriculture today, and many of the varieties of winter hardy alfalfa that have been introduced since 1953 were derived from vernal. So it's not the most common alfalfa used today, but it is the parent of a number of them that are still being used. As Brink looked back on this, he characterizes this as his principal direct contribution to agriculture. So I should just say a few words about Brink. He was just a charming, delightful man. When I came to the University of Wisconsin as an assistant professor, he would often take me aside and talk to me about things like what should you be doing to raise funds to support your research, what should you be doing to contribute to the university, what's your philosophy of the commercial development of products that come out of university research. Brink very much believed that the university had an obligation to do what it could to improve agriculture and medicine, and whatever other industrial uses knowledge could be put to. He thought that the university was here to help mankind. On the other hand, he didn't think that university professors were here to get rich off of what they did. And he was quite resistant to the idea of developing commercial products in a university laboratory or a university experimental farm. He thought that once you had established the methods and once you'd done the feasibility study, that you ought to turn it over to private industry to develop the product. And that product development wasn't the job of a university. So Brink didn't become rich as a result of developing hybrid corn that would grow in abundance in Wisconsin and make a lot of money for the seed companies. He didn't become rich off of alfalfa or sweet clover, but he was very proud of the contributions that he made in the development of these crops. One of his other do-good projects was that during the Great Depression, he realized that there were a lot of farms in Wisconsin that couldn't really grow alfalfa very well because their soil was too acid. And they needed treatment with agricultural lime, which is crushed limestone or something called marl, or the throwaway products from paper mills. Anyway, there are a number of things that can be used to spread on the fields to buffer them and bring their pH up. And he suggested that there was a federal program, the Federal Employment Relief Program, that provided money for hiring people who were out of work. This was the depths of the Depression, and there were millions of people who needed jobs in the United States. And Brink thought that one of the things that the university could do would be to organize a lime production program in the state of Wisconsin for the liming of agricultural farms that were trying to grow alfalfa. And he went to the dean of College of Agriculture and said, "How about this? What do you think?" And the dean talked to the head of agricultural extension, who said, there are going to be commercial companies out there that produce lime that are not going to like this. I don't want to rock the boat. And Brink, as he was wont to do, said, well, the heck with that. He went to talk to a friend of his who was the head of the Wisconsin Unemployment Compensation Program, Paul Rauschenbusch, and said, what do you think? Can't we create a program like this outside the university? And Rauschenbusch said, yeah, I think we could. He went to talk to legislators and got them to pass a bill that enabled county boards to set up programs. So the university didn't have to do it, the state didn't do it, the county boards could do it. And it turned out that this program got $2 million in funds from the Federal Employment Relief Program, sold the lime at cost to farmers who paid a total of $2 million into the program to get their lime, which helped to cover the cost of the people who were hired to produce the lime. And it provided jobs for 10,000 out-of-work men during the depths of the Depression. It was men. I mean, it specifically talked about men. So I suppose women could have got out and dug lime and crushed it, but for some reason this program was designed that way. Times have changed. Now let me talk a little bit about Brink's basic research. He was very much interested, as I said, in research that was of practical value, but he also did a lot of things that were just purely basic research driven by curiosity. His first project, as I said, in East's lab was to work on the physiology of pollen, growth physiology outside of a plant, so that you could treat it with various chemicals, adjust its nutrition, and see what was required for pollen growth. He studied the role of seed endosperm, that's all of the stuff outside of the embryo in a seed, in the development of a seed and in the development of an embryo. He studied unstable genes, and I'll tell you a little bit about that, and transposable elements. So he was one of the discoverers of transposable elements, or jumping genes, as you probably know. He studied a kind of non-Mendelian inheritance called paramutation that I want to tell you about. And he was interested in phase change during development in plants. He realized that all of the cells of a plant have the same chromosomal same chromosomal complement in them, but they don't all act the same way. That sometimes cells will suddenly switch from one mode of existence to another. For example, that sometimes you can take a cutting of a stem and stick it in the ground and it'll suddenly spring roots and put out leaf buds and grow into an adult plant, whereas it was just a differentiated stem before you started. So what's different? I mean, it's the same question that you get in any developmental system where cells that are doing one thing suddenly become able to do different things from other cells on the same organism. In the case of plants, it was often possible for differentiated cells to de-differentiate, so you could start off with adult cells and suddenly produce embryonic cells and make a new plant from them. So he was interested in that problem and wrote a long review article on the subject that stimulated a lot of research. Let me talk a little bit about transposable elements. This is a person who's famous for the discovery of transposable elements, Barbara McClintock, and she won the Nobel Prize in 1983 for her work. But at the same time that Barbara McClintock was doing work at Cold Spring Harbor in New York on unstable genes and transposable elements, Brink was doing similar work, almost identical work, here at the University of Wisconsin. And his system was simpler, actually, than Barbara McClintock's system. And the papers that he published were more understandable than Barbara McClintock's papers. Barbara McClintock was brilliant and sort of famously incomprehensible. It took a lot of effort to understand what Barbara McClintock was saying when she presented her scientific data. So if Brink did nothing else, he published papers that not only confirmed her results, but also were understandable to ordinary readers. When she won the Nobel Prize, I asked Brink what he thought about Barbara McClintock's getting the prize and his getting nothing. She was one of those one-person prizes. She got the whole thing. And they could have given it to Brink too, but he said no, it was perfectly right that that Barbara got there first and that she deserved recognition. Although his discovery of transposable elements was independent of Barbara's. So here's what they worked with. Here's a specimen of Barbara McClintock's corn. And here are the phenotypes of these kernels on the corn. Some kernels were red, and some kernels were colorless, and some kernels looked like this. They were variegated. And the question was, what was that all about? And what was known, other people had seen these things before. Emerson, for example, had seen these things before. And had realized that this meant that there was an unstable mutation in corn that during the development of a corn kernel underwent reversion to wild type, so that some cells in the corn had the normal gene for red pigment in them, and they could make red pigment against a background of mutant white cells. And the question was, since usually this didn't happen in the normal mutation to colorless pericarp, as it's called, the layer here that's got the pigment in it is pericarp, what was causing these guys to revert with such high frequency to wild type? And Brink studied exactly the same phenomenon, and here's a picture of Brink's picture of his phenotypes. Here is the red pericarp variety. Here is the highly unstable variety, and here is a less unstable variety. But both of these guys are variegated. This is heavily variegated. This is slightly variegated. And Brink, it's too much to try to follow this experiment for you, but I can tell you what they concluded. In every one of these corn cobs, one parent contributed a chromosome that had a mutation in the red gene that made it completely inactive. So that's just a normal mutation. You change a nucleotide, or you take out a piece of the gene, or you take out a little chunk of gene, or whatever. So this is a completely non-functional allele of the gene. And this is the normal gene. And in every one of these three kernels, one of the parents carried this stable, colorless mutation. And the other one carried either this or this or this. This is a red gene that has a transposable element, a jumping gene, inserted into it. This is a chromosome that has a jumping gene stuck into the red chromosome, and another copy of the same gene at another location, often in the same chromosome as the red gene, but sometimes somewhere else. And it turns out that when you put these mutant genes into a corn kernel, that with a fairly high frequency, this yellow thing, this transposable element, can pop out of the gene and go somewhere else in the genome of the corn, and return this variegated looking gene back to this form, so that this morphology becomes this morphology. It just becomes a perfectly normal functional red gene. So in a rather large number of cells here, as these kernels developed, the modulator, as he called it, the transposable element, jumped out and went somewhere else and returned the gene to its wild type form. It turned out that the presence of an extra modulator in the cell suppressed that to some extent, so that although the modulator in the red gene was still capable of popping out and going somewhere else, it didn't do it as often. And that produced this weak variegated phenotype. So what I just said is this, that the red gene can undergo a mutation to a stable colorless form, or it can undergo mutation to variegated form in which it has this element stuck into it, but it's like the F factor in a bacterial chromosome. It's inserted itself into the gene, but it can pop out again and return the gene to its original red state. And these variegated cells, this variegated chromosome, can have another transposable element inserted into it, and then the modulator can still pop out of the mutant gene, but it pops out less often. And it turns out that these two types of mutations, I ran out of arrows in creating this slide, but these two kinds of mutations can occur at the same time. That these transposable elements can pop out of this gene and attach themselves somewhere else in the genome, so that this can return to a wild type form, and at the same time the modulator from this gene, which is popped out, can attach itself to a sister copy of that gene and produce a chromosome that contains two modulators, one in the red gene and one separate. This was the beginning of a whole huge research world in which we have discovered now that transposable elements are everywhere in the world. Eukaryotes have them all over. We have them. If you take the DNA out of our chromosomes, and there are 46 chromosomes per body cell in a human, and you take the DNA out and you put it all end to end, it's about 2 meters long from one cell. Of that 2 meters, about 3/4 is transposable elements. So our genome is just full of these things. Some of which can still pop around from one place to another, but most of which have lost their ability to transpose. But somehow we got them all scattered throughout our genome, and presumably it was through a process like this. Replication of transposable elements and insertion in new positions. So this is an important question in evolution. Where do these things come from, and what are they good for? Or are they good for anything at all? I want to talk a little bit about one other thing that Brink did. He studied a phenomenon that he called paramutation. And by the time he died, he really didn't know what paramutation was all about, but the phenomenon interested him. Let's see, can you see the colors here? No. Well, I can see the colors. Anyway, this guy is dark red and this is light red. This is dark red and this is light red. In a normal Mendelian inheritance pattern, if you cross a big red, big R plant with a little r, where this is a recessive mutation, all of the offspring are big R, little r offspring, and they all are bright red. If you cross that guy to a little r, which is light red, you get offspring that are half big R and half little r. And the big R's are red, deep red, and the little R's are light red. Brink noticed that sometimes when he did a cross like this, depending on what the little r was, he got a different pattern of inheritance. He'd cross a bright red one to a light red one, he'd get bright red offspring. He'd get bright red light red offspring. He'd cross them to big Rs, and he'd get, everybody was light red. The little r allele was capable of somehow turning off the gene expression of the big R gene. So that this was a process that Brink called paramutation, in which a paramutating mutation could turn off the function of a paramutable mutation in the same gene coming from another parent, and the expression of the big R gene was just completely turned off in the presence of the little r. However, it was the big R genes were still there. If you got them out of here, you crossed them back to big R's for many generations, and you extracted these big R genes, they could slowly over time resume their function and get back the big R phenotype. So there was something that was pretty powerful, that turned off gene function, but that wasn't permanent. It wasn't a Mendelian property in the sense that you got back the original genotype when you crossed these organisms to little r recessives. This was just a mysterious process at the time that Brink did the work. He knew that the genes were being turned off by paramutating alleles. He knew that you could get back the function if you waited long enough. He didn't know what the mechanism for that might be. Today, one of the hot topics in genetics is gene silencing. And gene silencing has a lot to do with methylation of DNA and with small RNA molecules that are called suppressor RNAs that are involved in gene regulation. And this phenomenon now is one of the hot things that's being talked about all over the place, but not called by paramutation. It's called instead gene silencing. And it's an important process, apparently, in development and in evolution. So Brink's interest in this subject showed his good taste in problems. He unfortunately didn't live long enough to solve this problem. Let me say some other things that tell you some other things that Brink did as contributions just to the world in general while he was here. He was one of the founders of the Genetic Society of America. He was chair of genetics from 1939 to 1951. He was president of the Genetic Society of America at one time and managing editor of their journal, Genetics, for 5 years. He was president of the American Society of Naturalists. He engineered the merger of our two genetics departments. I told you last time when Josh Letterberg had been here for 10 years, he decided that we should have a department of medical genetics because the department in the College of Agriculture wasn't doing enough research that he thought was relevant to medicine. And he founded a department of medical genetics, and then because of the administrative structure of the university, these two departments had to operate more or less independently. When I first came here, there were two departments, and we sort of met from time to time and talked about our mutual problems, but we had to go back into separate sessions to do any formal business. Votes taken in the meeting of the two departments together were not binding. Only binding actions could be taken were taken in separate meetings. Brink said, "This is silly. We ought to act as if we're one department." And he engineered the formation of the Laboratory of Genetics in 1965, which put the two departments together. We now operate with one executive committee. When we hire new people, we hire them as one department. We make all of our decisions about undergraduate and graduate curriculum as one department and so forth. He was very proud of that accomplishment, and it was tricky because the deans didn't like the idea of having two departments operate as if they were one. They didn't want, the med school dean didn't want the College of Agriculture to affect medical school programs and vice versa. Brink was a member of the National Academy of Science, a high honor for any scientist, and a member of the American Academy of Arts and Sciences, another high honor. Just one final word. On the last day of his life, in October of 1984, Brink spent his morning in his cornfield. He was still doing research right up to the end, and he was in the process of trying to breed corn that would make a useful additive to silage, to alfalfa silage, to improve the protein quality of the silage. And there he was out in his field on October 2nd, 1984. Then he went home and died. He was an extraordinary person, and I'm glad to have had an opportunity to tell you about it.
I have a question.
Yeah.
When he was doing his initial studies on corn, was he increasing hardiness and interbreeding and hybridization? Was he aware of ploidy and like the changing in chromosome numbers, or was he just kind of like playing with plants and seeing like he's getting one that's bigger?
Yeah, people knew about ploidy then.
Yeah.
Yes,
okay.
I don't know that he was trying to do anything about the ploidy of corn at the time when he was doing his breeding experiments, but he was, and he was involved in lots of cytogenetic research. So he worked with Delmer Cooper, who was another professor in our department, on chromosomal issues. So he was acutely aware of chromosome numbers and chromosome structure. Any other thoughts, comments, questions? Well, thanks.